Students, welcome back. A biology bridge course, first PUC, Non Bosco PU College, Chitradurga. Myself, Abdul Wahab, biology lecturer. In the previous class, we discussed the a first PUC biology bridge course, a chapter called Digestion and Absorption. In this topic, in this chapter, we discussed the what is meant by digestion, what are the food and its components like carbohydrates, lipids, proteins, vitamins, minerals, water, etc. Lipids and carbohydrates are called as energy producers. Proteins are called as body builders. Water, mineral and vitamins are called as a body regulators. Then in the previous class we discussed the digestive system. The human digestive system consists of two components. One is the alimentary canal, another called as a digestive glands. Alimentary canal, how the food is passes from one part to the another part of the alimentary canal. So the alimentary canal or digestive system is starts from mouth and ends with anus. The mouth, first part, the food able to enter. Then the food is passed into pharynx, a common passage, esophagus, a food pipe. Between the esophagus and pharynx, there is a one lid-like structure called epiglottis. It serves the opening and closing of the esophagus as well as the trachea with respect to the food and air. Then the food is enter into the stomach. Here the food is stored for some duration. Here the chemical as well as mechanical digestion is taken place. After that the food is become semi-solid type called as a chyme, enter into the a first part of the small intestine called as a duodenum. The small intestine having a three parts: first duodenum, middle jejunum, last ileum. The site of absorption is the nothing but the the site of absorption and digestion nothing but the small intestine. Most of the food molecule able to digest or break down in the small intestine as well as absorption process also taken place. Finally, the undigested food is enter into the large intestine. The large intestine reabsorb the essential material as well as excess amount of water from the large intestine. The large intestine leads a storehouse of undigested food molecule called as a rectum. Rectum having a opening, that opening is called as a anus. By this anus the food, undigested food is goes outside. Now in this class we have discussed the second component of the digestive system called as digestive glands, and next is the how the chemical digestion process is taken place in the human body. First of all, digestive glands. The glands, nothing but the secreting structures. The digestive glands majorly exocrine glands, duct glands. They are able to secrete the chemical secretions called as enzymes. Those enzymes are helpful for the breakdown the organic food molecules like carbohydrate, proteins, lipids completely. Then the digestive glands are like, one is salivary gland, second one is the a liver, third one is the pancreas, gastric glands and intestinal glands. The salivary glands, there are three pairs of salivary glands like parotid glands, submaxillary glands, sublingual glands present in the buccal cavity or mouth region. The salivary glands able to secrete the a liquid called as a saliva. The saliva contains saliva or salivary juice. This saliva or salivary juice contains the one a significant enzyme called as a salivary amylase, also called as a ptyalin, Ptylin. This ptyalin is a carbohydrate breakdowning or digesting enzyme. One more enzyme is present called as a nothing but a lysozyme. It is an antibacterial enzyme. Next, the Next one is the a liver. liver. Liver is the second. Liver is the largest gland. Its weight is about 1.5 kilogram. The liver having a gallbladder. The liver able to secrete the juice called as a bile juice. That bile juice is stored in the gallbladder. Such gallbladder passes the bile juice into the duodenum of the small intestine by a duct called as a cystic duct. Liver is a chemical factory of our body because all the metabolism process, most of the metabolism process taken place in the liver. The liver secretes the bile juice. The bile juice does not contains any type of the digestive enzyme, but the bile juice is essential for the digestion of lipids by a mechanism called as a emulsification of fat. The bile juice contains bile salts and bile pigments. The bile pigments like bilirubin and biliverdin, and the bile salts like a sodium, potassium tarcholate and glycolate. These pigments as well as the salts helpful for the emulsification of fat. Emulsification of fat means that's just they are able to loosening the bonds inside the fat region. Okay. The next is called as a pancreas. It is the second largest gland present in the loop of the duodenum. So the pancreas is a mixed gland or dual gland or heterocrine gland. heterocrine gland. Why it is a mixed gland or heterocrine gland or dual gland? Because the pancreas perform both endocrine gland as well as a exocrine gland. It is able to secrete the enzymes as well as a hormones. So the two parts are there in the pancreas. One is the islets of Langerhans, another called the acinar cells. Acinar cells perform the exocrine gland which secrete the enzymes. The islets of Langerhans of pancreas act as a endocrine gland, secrete the hormones. In the islets of Langerhans, the alpha cell, beta cells as well as the delta cells are present. The alpha cells produces the glucagon as a hormone. The beta cells produces the insulin, and then a delta cell produces the somatostatin. The pancreas, the acinar cells of the pancreas produces the a juice called as a pancreatic juice. This pancreatic juice contains the pancreatic amylase as well as a pancreatic lipase. Pancreatic amylase is a carbohydrate digesting enzyme. Pancreatic lipase is a lipid digesting enzyme. Next, we are move on to the gastric gland. These gastric glands are present in the inner wall of the stomach. So, the gastric glands consist the majorly some cells like chief or peptic cells which secrete the proenzymes. Second, oxantic or parietal cells which secrete the HCl. The third one, a goblet cells or mucus cells which secrete the mucus. Okay, the gastric glands secrete the gastric juices. The gastric juice contains the two major enzymes like one is pepsin, other called as a renin. The pepsin is a protein digesting enzyme and the renin is a mammal's milk, specifically protein digesting enzyme. Okay. Next, intestinal glands. The intestinal glands present in the inner wall of the intestine, small intestine. The intestinal glands secrete the intestinal juice. That juice is called as a succus entericus. Succus entericus. There are two types of intestinal glands. Those are crypt of Lieberkühn and Brunner's gland. The intestinal gland, what it secretes the intestinal juice. Such juice contains the enzymes like enterokinase, also called as a enteropeptidase, amino peptidase, tripeptidase, dipeptidase, maltase, sucrase. Sucrase is also called as invertase. Finally, lactase. Okay. Then this is about the digestive system. Next, we are move on to the digestion. Exact the chemical digestion, how it is taken place. Digestion, we know that it is a physiological process of splitting the macromolecules non-diffusible food into micromolecular diffusible one, or the breakdown of complex food molecule into simple observable form. There are two types of digestion. One is a mechanical digestion, another is a chemical digestion. Mechanical digestion means it is a fragmentation of food by mechanical energy. It includes mastication, deglution, or swallowing. Then peristalsis. The chemical digestion, the process of fragmentation or breakdown of food by using enzyme. So, when come to the chemical digestion, first one, carbohydrate digestion. We are going to see majorly three type of digestion. First one is the carbohydrate digestion, second protein digestion, third a lipid digestion. When come to the carbohydrate digestion in the buccal cavity, first buccal cavity. In the buccal cavity, the salivary glands secrete the salivary amylase enzyme called or is also called as a ptyalin. This enzyme able to break down the carbohydrate into maltose. So in the buccal cavity, what happens when we consume the food? The food is cut into small pieces by chewing action of the jaws. Then reduction of large food particles into smaller particles with the help of teeth is called as a mastication. Then the tongue mixes the food well with the saliva. The process of mixing of food with the saliva is called ensalivation. So the carbohydration starts in the buccal cavity itself. The salivary gland secrete the salivary amylase enzyme called ptyalin. Such enzyme break down the carbohydrate like starch and convert it into maltose. Next, carbohydrate digestion in stomach. In the stomach, there is no any carbohydrate digestion is taken place. So there is a mechanical digestion called as a peristalsis is taken place in the esophagus as well as a stomach. Next, when come to the stomach, there is no any carbohydrate digestive enzyme. So there is no any carbohydrate digestion taken place in the stomach. When come to the intestine, the pancreatic amylase enzyme is also called as a amylopsin. This enzyme is converts the starch or glycogen into maltose. Then next, the intestinal enzymes like maltase, sucrase and lactase. Maltase able to convert the or digest the maltose as a disaccharide into a monosaccharides like glucose plus glucose. The lactase enzyme is convert into glucose plus galactose. Then the sucrase enzyme is convert the sucrose into glucose plus fructose. In this way, the carbohydrate, it is a polysaccharide, completely break down by an enzyme's list of enzyme. Finally, the end product like a glucose molecule occurs. Okay, this is a summary of carbohydrate digestion. Next, we are move on to the protein digestion. The protein digestion starts in stomach as well as intestine. There is no any digestion taken place in the mouth region because there is no any protein digesting enzymes are present. When come to the stomach, there are the gastric glands secrete a gastric juice which contains the protein digesting enzymes like pepsin and renin. Pepsin, it is an active form, but it is present in active form like a pepsinogen. The HCl responsible to convert the pepsinogen into pepsin. The stomach not only secrete the gastric juice, also secrete the HCl, hydrochloric acid. The HCl is helps the conversion of pepsinogen into pepsin. It helps to kills the some bacteria, disease causing bacteria entering with the food molecule. Then when the pepsin is formed, this pepsin able to break down or converts the proteins into proteases, peptones and polypeptides. The second enzyme called as a renin is also called as a rennet or chymosin. It is a milk curdling enzyme. It is a unique mammal's milk digesting enzyme. This enzyme able to act on protein digesting. The renin is present in active form called as a prorenin. This prorenin is converted into active renin by HCl. The renin acts on milk protein casein, converting it into para-casein. Para-casein. The casein, the renin converts the casein into para-casein. The protein, the casein is a milk protein. When the para-casein is formed, this para-casein is become calcium para-caseinate with the help of calcium ions. Finally, the calcium para-caseinate is acted upon by pepsin and is converted into proteases, peptones and polypeptides. Next, the protein digestion starts. The protein digestion process is continuous in the small intestine. Here what happens when the food is enter into the stomach? In the stomach, the food is digested. The digested food is become semi-solid or semi-liquid, that is called as a chyme. Such chyme food is enter into the small intestine. The first part of the small intestine called as a duodenum. Such a duodenum able to receive the chyme, a semi-solid food molecule. Then in the intestine, what happens? The intestinal juice as well as pancreatic juice secrete the list of enzymes. First of all, a pancreatic juice having one enzyme called as a enterokinase. This enterokinase is converts the trypsinogen into trypsin. Then the trypsin is converts the proteases and peptones into polypeptides. Then what the trypsin is there? This enzyme is converts the chymotrypsinogen into chymotrypsin. This chymotrypsin enzyme is again break down the proteases and plus peptones into polypeptides. After that, the same trypsin is again converts the pro-carboxypeptidase into carboxypeptidase. The carboxypeptidase enzyme converts the proteases and peptones into polypeptides, tripeptides and dipeptides. Okay. Finally, the amino peptidase. It is a intestinal enzyme which converts the proteases, peptones into tripeptides, dipeptides, amino acids. Then the tripeptidase enzyme converts the tripeptides into amino acids. Then the dipeptidase enzyme converts the dipeptides into amino acids. Here the proteins are the complex organic food molecule. When the enzymes are able to act on these proteins or break down these proteins, the protein gives us the end products called as a amino acids. In this way, the protein digestion is complete. Then the third one is called as a digestion of lipids. Digestion of lipids. In the oral cavity, in the buccal cavity, there is a lingual lipase. One enzyme is there. This lingual lipase enzyme is secreted by Ebner's glands of tongue. This enzyme is just very smaller amount is converts the dietary glycerides into fatty acids and monoglycerides. Only 3 to 4% of fat is digested in the oral cavity or mouth. Next, digestion of fat in the stomach. In the stomach, there is a one enzyme called a gastric lipase. This gastric lipase, along with lingual lipase, is small amountly converts the triglycerides into fatty acids and monoglycerides. Majorly, the fat is digested in the intestine, small intestine. Small intestine. In the small intestine, the liver secretes the bile juice. The bile juice contains the bile salts and bile pigments. Both are able to emulsify the fat. Emulsification means breaking of large fat drops into small droplets. Okay. Then what happens? The fat is become emulsified fat. Then such emulsified fat, on that fat, the colipase enzyme is acted. This colipase enzyme is break down the emulsified fat. Then what happens? There is a colipase enzymes. When it able to activate the emulsified fat, then the one more enzyme is formed called as a lipase enzyme. This lipase enzyme is completely break down the emulsified fat into fatty acid and glycerol. So when the fatty acid and glycerols are formed, in that the lipid digestion is completed. So one thing is nothing but the amino acids are the end products of proteins. The fatty acid and glycerols are the end products of fats and lipids. The glucose is the end products of carbohydrates. So in this way, the carbohydrates, proteins and lipids are able to digest it. When come to the absorption, in mouth, certain drug molecules contact with the mucosa of mouth and lower side of the tongue are absorbed into blood capillaries lining. In the stomach, absorption of water, simple sugars and alcohol etc. taken place. In the small intestine, the absorption of nutrients majorly occurs. The glucose, fructose, fatty acids, glycerol, amino acids are absorbed through the mucosa of small intestine. Then it is entered in the bloodstream and lymph. In the large intestine, absorption of excess amount of water and other essential materials. Then apart from some drug molecules are also taken place. The absorption of fatty acid and glycerol is somewhat complicated, because the fatty acid and glycerols being insoluble cannot be absorbed into the blood easily. They are first break down into small droplets. Those droplets are called as micelles, which move into the intestinal mucosa. In the intestinal mucosa, what happens? A very small protein coated fat globules are there called chylomicron. Those transport the micelles into lymph vessels in the villi. These lymph vessels ultimately release the absorbed substances into the blood stream. In this way, the absorption of fatty acid and glycerol is taken place. Okay. Then assimilation. It is a process in which the absorbed substance finally reach the tissue and utilize them for their activities. The last step is called as a defecation. The ejection of feces, excretory waste to outside through the anal opening is called as a defecation. This is about the first ingestion, intake of food, digestion, breakdown of food. Third, assimilation is nothing but the first ingestion, intake of food. Second, digestion, breakdown of food. Third, absorption, a blood able to absorb the nutrient. Next, assimilation, distribution of the essential materials each and every cells. Then defecation, the ejection of undigested food molecule. Then the disorders of digestive system. The disorders are like, one is jaundice, vomiting, diarrhea, constipation, indigestion. Jaundice is also called as icterus. It is caused by due to the abdominal bilirubin metabolism. So what happens? The liver cells are able to abnormally break down the RBCs. Then the abnormal production of bilirubin we are going to see. This bilirubin is able to deposit it here under the skin, under the eye sclera, or any other region. Those regions are look like a yellow. That condition is called as a jaundice. Second, vomiting. It is the ingestion of the stomach contents through the mouth. Okay. Diarrhea. The abdominal frequency of bowel movement and increased liquidity of the fecal discharge is known as a diarrhea. It reduces the absorption of the food. Constipation. The feces are retained within the rectum as the bowel movements occur irregularly. Then indigestion. The condition, the food is not properly digested, leading to the feeling of fullness. Such the causes of indigestion reduce the enzyme secretion, anxiety, food poisoning, overeating, spicy food. So these are the disorders of digestive system. So in this digestion and absorption chapter, I am going to give a brief concept of digestion absorption process. Okay, in the regular classes we are going to study in detail. Okay. This is the first bridge course, a chapter called as a digestion and absorption. Next class, one more bridge course chapter we are going to study it called as a respiration. Thank you.